I think you're muted, Dr. McCormick.
Okay, there we go. Hi, I'm David McCormick. I'm Director of the Institute of Neuroscience, and I'm your moderator today. So I'll be calling on each of you to introduce yourselves, talk about your research, and tell us what you did and what you found, and then we can have a discussion on it. So take the next minute or so for make sure your guests are here. Now, are they, some people had co-authors, like Dakota, your co-author is Nisha, correct? So Dakota will be presenting, I guess, because he has presenter on his.
McCormick, I am having a very hard time hearing you. I'm not sure if anyone else is.
Anybody else having a hard time hearing you?
Okay. We're getting some. It might be me, but my volume's almost all the way up and I am not hearing you well.
You hear me now?
Much better. Yes.
All right. You need a podcast microphone to do
Yes, I love it. Very professional.
this. Okay, as I was saying, we'll go in order of the authors on the program. I'll give you a few minutes to present your research. You can share your screen if you like and show your poster. I've watched your videos. I'm sure you all watched your, hopefully other people's videos also of their posters on the YouTube channel. And, you know, it's hard to go through the whole poster, just 3 or 4 minutes, but now's your chance to tell us about your research, and then we'll leave a good 10 or 15 minutes at the end for question and answers. All right. So Emmalyn, you're here. I see you.
Hello.
You can, let's see, Paige, I guess they have the ability to share their screen if they like.
It looks like I do, yeah.
Okay, great. Emmalyn, why don't you tell
Can
us
you
about
see
your
my-
research?
Can you see my poster?
Yep, I sure do.
Great. Okay, so firstly, I want to say that this may sound a little bit like a project proposal. And that's because although I have collected preliminary data at this point and spent a lot of time developing methodology protocols and all that, the methods of this project are very labor intensive and actually require much more troubleshooting than I initially assumed, which I think is always the case. But anyways, so as a result, I have not completed, many aspects of this project. So today I'm going to mainly present the background of my research topic and then speak about the methodology of my project that I have been developing and practicing since I joined the lab of Christopher Neal in the Institute of Neuroscience about 2 years ago. So the topic of my research is visual attentional control, and that's a behavioral process that is absolutely critical for survival across all species that include vision. And as we know, as humans, we use vision. So if we were to simply view all objects in our visual field with both equal focus and attention, our mind would probably be far too overwhelmed to make good meaning of it all. So therefore, there exists an array of brain mechanisms that we utilize, some conscious and others not, that allow us to sort of selectively pay attention to objects that we deem important for our survival. So objects considered important are things that we may need to react to, and as such, that is what is in our focus. So at the very bottom here, can you see my pointer? So at the very bottom
Yeah,
here,
we can see.
we've got, you know, a picture of a busy street, and there is a lot to navigate, right? There's bikes, crowds of people, and probably things like buildings and cars. So any particular person in this image is probably focusing their eyesight on potential obstacles, while simultaneously ignoring the objects that they consider unimportant, which are things like, you know, the trees and the street signs. So we maximize our safety and thus our chance of survival when we employ this selective attention while viewing the world around us. The biological mechanism of this seemingly simple process, which I call visual attentional control, actually remains somewhat of a mystery today, despite its importance because of the historical inability to measure and control brain activity across all organisms. This project is based on my attempt to begin to reveal a possible mechanism of visual attentional control through the utilization of the mouse model organism, whose visual system structure and function is highly analogous to that of humans. It's just, you know, a whole lot smaller. So the central pathway followed for visual perception to occur is pretty well known, despite the complexity and variety of processes that involve, you know, all aspects of our vision. Visual information enters the eye, and then it hits photoreceptors in the back of the retina, or in the back of the eye at the retina, which then sends information along the optic nerve into the center of the brain. In the center of the brain is the thalamus, which serves as a relay center for sensory information. So the thalamus receives signals of everything that is perceived by the eye, and then it sorts through various components of the signal and sends the information off to multiple other areas outside of the thalamus that achieve higher order processing necessary for accurate perception. So what I mean by higher order processing is that, for example, the top-down processes that we use, such as visual memory, object recognition, and tuning to object orientation. So one subregion of the thalamus heavily involved in vision in this pathway is called the LGN, which is shown here in this, I'll zoom in a little bit, shown here in this pathway, connected to the eye. The LGN relays information up to the primary visual cortex, which is a structure involved with developing our basic perception of object contours in our visual field. Here, and also in this presentation, I'm going to refer to primary visual cortex as V1. So where things start to get more mysterious is how this information goes from V1 to higher order processing areas that allow us to see both accurately and with a greater level of detail. So while V1 sends some information directly up to those higher order processing areas, there is evidence of another pathway in which the information is sent once again back to the thalamus, this time to a different region, where unimportant information, visual information, such as, you know, the trees in the example, is filtered out as to not waste brain power. So after this filtration, information would then be sent back to V1 and then relayed to higher order processing areas. The subregion of the thalamus implicated in this process is called the pulvinar, this green mass here. And historically, the pulvinar has remained highly enigmatic in function. Interestingly, the pulvinar has been found to receive feedback from areas of the brain that are not involved with perception per se, but are instead involved with attention, such as the superior colliculus. This suggests that the filtration process that may be occurring in the pulvinar is modulated by the information it receives from these attentional control areas, basically telling it which information should be ignored and which should be attended to.
Emma, I want to interrupt real quick and say you only have like 3 more minutes, so you should speed it up and get to the.
Or are these timed?
Yeah, because we only have an hour. We have to move on to the next session.
Okay, the emails did not make that super clear. All right, that's fine.
Yeah.
So basically, mice can be trained to perform a task that tests their ability to do this. And so I'll present the mouse as shown in the methods here with a visual stimulus on a computer screen, and they will have to report changes in the orientation to the stimulus by licking a water port. So they are going to be trained on this task, and they will have a water restrictive diet, which helps drive their motivation. And when they incorrectly report a change, they will receive an air puff instead, which discourages them from misreporting again. So once they are sufficiently trained, I will shut down activity in the pulvinar using this system called DREADS. I will introduce it with a virus, and it basically introduces a receptor onto the cells in the pulvinar that when I inject a special drug called CNO, the cells become inactive. And the activity from the pulvinar cells are inhibited entirely, theoretically. So this results in a temporary, not permanent shutdown of pulvinar activity until the clozapine or the CNO becomes fully metabolized. So simultaneously, I'm also going to record activity in V1 through cranial window, which is shown in the area here. It's a surgical procedure. And in order to visualize activity, I tag the pulvinar cells with a green fluorescent protein called GCAMP. And that's actually the tag shown here in A. So cells based in the pulvinar have lots of projection spreading all the way to V1. And by only tagging pulvinar cells and recording from V1, I can see how the pulvinar originating activity in V1 differs when the animal is actively taking part in behavior versus when it is passively, viewing a stimulus. And I can also correlate brain activity with things like the visual stimulus, movement, and orientation. So in the setup here, you can see that they are positioned for recording simultaneously. So basically, I expect to see, firstly, to find that behavioral task performance is significantly worse in mice that have had their pulvinar silenced by the DREAD system compared to ones that have not. And I find that recorded activity in V1 via the two-photon microscopy from the pulvinar will show a much higher intensity when the mice are taking part in the active visual task, as opposed to when they are passively viewing the stimulus. So during shutdown experiments, I will also utilize this imaging technique to ensure that there's a proper shutdown and thus not that much GCAMP fluorescent changes to record. So far I've been able to show in the mapping images here that the connection between pulvinar, the axon projections between the pulvinar and V1 are sufficient to produce a retinotopic response in V1, meaning the response shows both temporal and spatial mapping in a topographic fashion. So the larger images here are examples of the type of two-photon data I will collect. I have not yet analyzed data that is axon-based, so these are actually little cell bodies, but that is a next step I will take once I gain access back to the lab. So analysis of two-photon images will be based around the pulling out of axons in the field of recording and comparing the cycles of activation and inactivation with the simultaneous cycling of visual stimulus. To finish up, I want to thank Dr. Chris Neal and Dr. Philip Parker for their support as I've navigated this extremely bumpy process. And I also want to thank the Center for Undergraduate Research and Engagement for providing me with a SIR fellowship for the 2019 summer. So that's about it. And I'm happy to hear all constructive feedback and criticism and any questions you may have.
Any quick questions?
I had a question. So about the filtering of through V1. So traditionally, or in cats at least, Hebel-Wiesel characterizations are like light-dark boundaries in V1. But you were saying something about trees. Is the pathway through more complex, or is there any idea about how a higher level order?
No, for the most part, you're right. Sorry if that was unclear. When I'm referring to the trees, I'm talking about a different process by which, after that initial perception, your brain, picks out, not to focus on the trees, but instead to focus on the people or something like that. Does that make sense? Maybe I don't totally understand your question.
Sure. No, I was just wondering if that was it for the system because if you have such simple input data from V1, how are you filtering? What kind of role of filtering that the pulvinar would be doing? I guess was just my question about clarifying that, because if it was, an abstract filter having to do with characterization, like a tree would be filtered, that makes sense. But if it's some level of filtering in light dark boundaries, that doesn't maybe is not quite as clear. So is there any clarification on how you could filter something that was maybe a little bit simpler classification based on boundaries, or am I mistaking the circuitry?
No, you're right. And it's obviously a lot more complicated than the simple scheme I have shown. V1 is not just a standalone structure that holds, connections to higher order processing areas, blah, blah, blah. So I think that there's actually a lot of input it's receiving from all sorts of areas of the brain, all sorts of areas of the cortex too. And that feedback between the thalamus and V1 is bidirectional. So you're right, V1 does kind of have the basic light, dark contour. That's the process that I think that we oftentimes we think that it's involved in, but I'm sure there's feedback coming from you have a full perception of a tree and then you decide that it's not worth attending to. I think that those higher order processing areas are not completely shut off until the filtering happens.
All right, well, thank you very much for an excellent presentation, Emmalyn. And we're going to move on now to Dakota Paulus. Dakota, you want to tell us about your poster and your findings?
Sure. So I just had a quick question. So I normally have this laid out to present with co-authors. Am I going to be the only one presenting, or is that format fine?
That's fine. You have about 10 minutes so that we don't run out of time for the other people.
Great. Okay, so
I can start.
yeah, go ahead.
Perfect. Okay, so hi, my name is Nisha, and I presented alongside Dakota and Katia Pramono, and our study is titled, actually I'm going to share my screen really quick, sorry. Okay. Okay, there we go. And our study is titled Feedback-Related Negativity as a Neural Indicator of Executive Function in Preschoolers. And this study is kind of similar to Emmalyn, where this is more of like a project proposal. We haven't been able to conduct this yet, but this is kind of an idea of what we'd like to do once restrictions on human subject research has been lifted. And this study is a part of the Observing Responsive Caregiving and Action Monitoring Project. We are we're all research assistants at the Stress Neurobiology and Prevention Lab.
So the general overarching purpose of our study is to observe a relationship between executive function and feedback related negativity. I'll go into that a little bit more, or we will in a little while here. But overall, executive function is a critical cognitive skill that supports early learning and development. It's influenced both genetically and through environmental factors. And it is very, it's hypothesized to be very concentrated in the prefrontal cortex, and more specifically, some of the other regions within that, like the dorsal anterior cingulate cortex. And so we're looking at executive function and its relationship to feedback-related negativity, which is a electrical impulse that's generated in the brain following positive or negative feedback to a specific stimulus. And our hope, once we are able to collect data, is to find a correlation with feedback-related negativity signals and the development of executive function that from data that we have gathered in the previous study that our lab has conducted. So, go ahead, Cody.
Cool. So like Dakota said, we're studying executive function, which is just a set of cognitive skills that helps a child regulate their behavior. And so the executive function of our participants was measured in the earlier part of our study using a task called EF Touch, where basically the participant will press answers on a tablet and they will be read the questions by a research assistant. And then also feedback-related negativity is, it's seen in EEG data as a negative deflection, and it happens following negative feedback. So it's generated by the prefrontal cortex, or more specifically, the dorsal anterior cingulate cortex, and we expect to observe this event-related potential as a response to negative feedback. So in this case, it's going to look like not getting a snack in a task, but we're going to explain more of our task later on.
So what we use to measure feedback-related negativity is a device known as an electroencephalogram, commonly known as EEG, the abbreviation. An electroencephalogram is essentially a device that measures the electrical brain activity of a participant. And in our study, we placed an electrode net on the child participant's head, and their brain activity was recorded as they completed a task. Electroencephalogram was particularly useful in our study because it gives a precise recording of when a reaction to a stimulus occurs. So it is especially useful when reading feedback-related negativity and recording that. So we performed a literature search prior to designing the study, and that's kind of what brought us to this kind of question. And we included some of the key studies that were particularly influential in designing our study. And what we've kind of found from our literature search is that research suggests that feedback-related negativity emerges as early as 2.5 years old, so even in toddlers. And in the studies we found, feedback-related negativity is greater in children compared to older study participants. And overall, we found that there's limited research studying feedback processing in early childhood. So we hope that our study can help contribute to this area of research as it develops more.
So the way that we are going to actually conduct this is by using a task called the Doors game, or the Doors task. And it's going to help us record feedback-related negativity by, it's going to be displaying two doors on a computer screen, and the child is going to have a controller in which they can pick between both of the doors. And once they click the button, there will either be a thumbs up or a thumbs down. And if there's a thumbs up, then the child will receive a snack, whatever the favorite that child chose to pick. And it will be immediately placed in a container in front of the child as to kind of help for motivation and keep the child engaged, getting that immediate reward there for getting the thumbs up. And if the child receives a thumbs down, they don't get one, and then the doors will be reset and go on to another trial. And while this is happening, when the thumbs up or thumbs down appears on the screen, it's time-stamped on our EEG data. That way we can see exactly when they're getting that feedback and be looking for those markers on the data that is showing the feedback-related negativity that we are looking for, hopefully. So yeah, that, and then we will be also comparing that data to other tasks, like I said, that show data for executive function, and doing a comparison of those results and hopefully seeing a positive correlation there.
So I'm going to go a little bit deeper into our methods here. So we're collecting data from 30 participants ages 4 to 6. And these participants have completed tasks during previous part of the observing responsive caregiving and action monitoring study. And so the sessions that we're running will run about 1 hour, and the child will just play the Doors task while wearing an EEG cap.
Okay, I think we should open it up to any quick questions before we go to the next presentation.
Sure, this is Tyson Barker. A quick question. Well, first, thank you for the presentation. I think this is really great. Generally, why does it matter? Like, why does it, with feedback, how kids' brains respond to feedback be important to exploring in terms of their development?
Yeah, thank you for your question. So overall, the way that children respond to feedback is really important because you want to make sure that everything that you do is helping to encourage that growth of executive function, because it's so important to other cognitive behaviors. Yeah, if the way that we understand FRN is helpful to understanding how to better promote positive reactions in kids, it's going to affect teachers and parents and everyone who interacts with children.
Awesome, thank you.
All right, I think we have to move on. Thank you very much, Dakota, Katya, and Nisha for a great presentation. Vanessa Hufnagel, you're up next.
Sounds good. Let me get the sharing screen thing together. Oh, maybe Nisha has to stop first.
Sorry about that. One sec.
Okay, perfect. Are you guys seeing my screen?
Yes.
Awesome. So before I start, I want to thank my research mentors, Alex Rockhill and Dr. Nicole Swan, for helping me out throughout this project. This was my honors college undergraduate thesis, and this wouldn't have been possible without them. So today I'll be presenting my study, Electrophysiological Patterns of Skilled Motor Movements. As the title states, we examine skilled movements in healthy subjects. Skill can be somewhat of a vague term and changes between disciplines, but in neuroscience and skill-related research, a movement pattern is considered skilled when processing of the environment and internally generated cues become refined. When you perform a task you're good at, adaptive strategies develop and task-relevant stimuli are suppressed. Research has been able to identify what makes the processing of a skilled movement different from other motor patterns. Experts exhibit less overall coherence, or in other words, communication between different areas of the brain, and cortical activation in the sensorimotor cortex relative to novices. In a sense, your brain doesn't have to work as hard to do the things that you're good at. Our study looked specifically at beta band frequency oscillations, which are associated with motor planning, execution, and inhibition. These oscillations are more commonly referred to as brain waves, and we can record these electrical signals non-invasively using electroencephalography, also called EEG. In performing this study, we didn't just look at skilled research findings. We also looked at research from a specific movement disorder called focal task-specific dystonia, abbreviated FTSD for short. This disorder affects a once refined movement and causes abnormal contractions in the muscles associated with the movement. These symptoms are only present when the affected person tries to perform the specific dexterous task. Because the disorder affects an overtrained skilled movement, there is a potential connection here to skilled-related research. Patients with FTSD exhibit a reduction in beta functional connectivity in sensorimotor areas. More so, these patients report greater variability in response time between sequential responses, but show no difference in error rates. With this in mind, the purpose of our study was to measure fluctuations in oscillatory activity during a familiar task condition and a foreign task condition. We wanted to investigate whether similar beta frequency signatures and response patterns seen in dystonic patients may be present in healthy people while performing familiar or skilled movements. So we developed 5 hypotheses to address our research questions. First, we anticipated we would see no difference in correct responses between trial types. Second, we expected to see greater variability of response times during the skilled trial type. Third, we anticipated reduced fluctuations in sensorimotor beta power during the skilled compared to the unskilled trial types. Here, we were more concerned with beta desynchronization and rebound. Beta desynchronization is a decrease in overall beta oscillatory power that occurs during movement planning and initiation. Beta rebound is the opposite. It is an increase in oscillatory beta power that occurs once movement has been terminated. Next, because of the timing of the trials and stimuli, we expected to see 4 separate beta power decreases and rebounds within a trial, 1 for each character of the sequence, and I'll get a little more into the methods in just a moment. And finally, for our 5th hypothesis, we expected to see reduced fluctuations in sensorimotor beta power over all trial types for people who are more proficient at typing. So we sought to test these hypotheses by using a keyboard typing task consisting of 2 different trial versions designed to emulate a more familiar task and a foreign task. Participants either saw a string of 4 numbers or a string of 4 letters as a stimulus in each trial. When participants encountered a letter stimulus, they typed the corresponding letter as usual using standard typing techniques. This was considered the skilled trial version. When participants encountered a number stimulus, they used a finger that corresponded to a specific number predetermined with the subject before the start of the experiment. This condition was thought to be less practiced, so it was considered the unskilled condition. If you look at the bottom left-hand side of the poster, there is a schematic that is color-coded with the fingers and then the keys that participants use to respond, and then underneath are the progressions of the skilled trials and the unskilled trials. Our behavioral findings are outlined in the topmost set of figures. From left to right, we have the response variability from the first responses, response variability for the second through 4th responses, and the percentage of key presses counted as correct. So we saw no significant difference in error rates between trial types. Variability in the first key press time also showed no significant difference. However, variability between 2nd, 3rd, and 4th responses were significantly greater during these unskilled trial types, which was unexpected as we actually anticipated seeing an increase in variability in the skilled trial types. The figures below these behavioral plots are spectrogram plots aligned to 3 major events in each of the trials. The first row of spectrograms are time-locked to the trial start. The second row of spectrograms are time-locked to the first movement or the first key press of the trial. And then the third row is time-locked to the fourth key press or movement offset. Each series of 3 spectrograms show the spectral power density from 2.5 Hertz to 64 Hertz. We're mainly interested in the activity between 13 and 30 Hertz because this is the beta frequency range. There is a spectrogram within each set, as denoted by the letters, for skilled movement, unskilled movement, and then what we're more interested in, because it shows the difference between the two, the values of skilled minus the unskilled values, and these are outlined in the red rectangle. We found there was no significant difference in beta desynchronization between trial types. We did, however, see decreased beta power driven by a weaker beta rebound after the fourth key press in skilled trial types. These are denoted, if you look at the third row here of the spectrograms, by the black arrow. This decrease in skilled beta power is consistent with EEG signatures observed in patients with FTSD. Addressing our fourth hypothesis, our results more closely resembled a continuous reduction in beta power throughout the trial. This may infer that participants did not observe each stimulus individually, but rather observed the string of stimuli prior to making their first response and coded the entire trial as one motor command. And our final EEG analysis compared sensory-motor beta power fluctuations to participant typing proficiency. The figures depicting these correlations are here on the bottom. Each graph uses the same typing assessment data, but beta power is divided by the electrode we sampled from and trial type. We used beta power from the fourth response, as this proved to be the most significant between trial types. Here, there were no significant correlations observed in skilled or unskilled beta power values and typing ability on either side of the sensory motor cortex. In summary, our study preliminarily demonstrates that the beta rebound period of the skilled trials have similar patterns to that of dystonic patients. Additionally, some, but not all, of our behavioral observations agreed with dystonic literature. In the future, we want to perform a cluster-based correction analysis to determine whether our results would survive a multiple comparisons test. As of now, our data shows the need to continue to investigate the relationship between skilled movement in healthy people and focal task-specific dystonia.
Great. Any quick questions for Vanessa?
I can ask a quick question. Can you hear me?
Yes.
Really nice job, Vanessa. I was just wondering if you could clarify why in your behavioral results you separated out the first from the second through fourth response in your analysis?
Yeah, absolutely. So there was an increased latency between the first as compared to the second, third, and fourth. So on average, it took subjects maybe a second to a second and a half to respond to the first stimulus, and then the consecutive ones were anywhere below half a second. And this finding kind of resonates what we had seen in the continuous reduction in beta power. So participants took the second before they responded to process the entirety of the stimuli that they were seeing, and then just responded one right after the other rather than processing them individually. So just because the values were so incredibly different, I did get a little off track there, sorry about that, but because the values were so incredibly different, we thought it'd be best to analyze these separately.
Thanks. Good job.
All right. Well, thank you very much for your wonderful presentation. Raj Shah, or I see Hetal Shah on the
Oh no, yeah,
screen
it's
here.
me right here. I'm gonna present.
What name do you go by?
Raj.
Raj. Okay, great.
Yeah, it's just Raj.
Take it away.
Right. Okay, I'm sharing screen. Can everyone see the screen? Cool. Hope everyone is well. My name is Raj Shah. Before I begin, I would like to thank my mentors, Santiago Jaramillo, as well as Beth McCary, and fellow undergraduate researcher Bridget Deck for all their support in this project. Motivation. How does the brain process sounds? In order to understand this, we have a powerful tool known as two-photon calcium imaging that allows us to measure the activity of hundreds of individual neurons simultaneously. As you can see in figure 1, we can do this in vivo, which means while the mouse is awake, we're able to present sounds and image the neurons that are related to auditory processing. Methods. in order for the microscope to have visual access to the brain, you can see in figure 2 that we replace the piece of skull that is above the cortex with a glass window. So now the two-photon microscope has direct visual access to the cortex. So how does this calcium imaging actually measure the activity of neurons? So since the concentration of calcium in a cell is directly correlated with the level of neural activity, we have a fluorescent calcium sensor known as GCamp that allows us to measure the activity of these neurons based on the level of fluorescence. Surgery. So in order to gain optical access to the auditory cortex for use in this type of imaging process, it needs to be required through surgical intervention. And in figure 3, you can kind of see a basic sketch of how this surgery is done. So we first dissect the scalp to expose the skull, and then we mark the coordinates of the auditory cortex using a drill. And then we make a 3mm window and replace that piece of skull with a glass window. There was many challenges associated with this surgery. More specifically, since the auditory cortex is on like the lateral aspect of the brain, due to the surrounding large muscles and arteries around the ears, it's not very easily accessible. So we actually did a partial dissection of temporal muscle, which allowed for increased access and also minimized blood loss. Also, due to the inflammation and pain caused by the invasiveness of this surgery, the drug protocol was modified that allowed for an optimizing of the process as well as increase in survival rates. You can see in figure 4, once a mouse is successfully implanted with a cranial window, you'll be able to see the arteries of the brain directly through the window. Imaging. So in figure 5, you can see that once a mouse is head fixed and running on a wheel, we're able to use a two-photon microscope to directly image the auditory cortex using a cranial window. In figure 6, you can see in this calcium imaging that as the neuron's fluorescence increases, that's an indicator of the increase of concentration of calcium in a cell. And since that is a reliable indicator of neural activity, we can start using GCamp in combination with this two-photon imaging to measure these activities of neurons in the auditory cortex. Results. So by overcoming the challenges of surgery, we were able to successfully implant this cranial window over the auditory cortex, which has allowed us to use this two-photon calcium imaging to measure the activity of neurons in the auditory cortex while the mouse is awake and being presented with sounds. Future directions. So we now have access to the auditory cortex and we're allowed to image. So now we wanted to see how our ability to detect changes in sound is dependent on the activation of neurons in the auditory cortex. So you can see in figure 7 that when we set up a task that requires a mouse to process changes in sound, we can use this two-photon imaging to see if our ability to detect changes in sound is dependent on the activation of neurons in the auditory cortex. This will allow us to further better understand how the brain processes sounds. Acknowledgments, I would like to thank the Jaramillo lab members for supporting me through this project and being a real big family throughout the way, Terrestrial Animal Care Services, as well as the Institute of Neuroscience for all their support. Thank you. Any questions?
Okay, great. Any questions? I have a question for you. Since the auditory cortex is so far lateral, did you have a microscope that the objective could come to the side, or did you tilt the animal's head like this?
We had like the angles measured out so we had the microscope come at an angle, so it was able to directly kind of see the cranial window exactly. So every time he was head fixed on the wheel, the microscope would directly be over the auditory cortex window.
The mouse could be comfortable and have his head straight.
Correct.
Great. And what layer were you able to image with the microscope?
So that was the plan during spring term, that we were going to kind of do more imaging once we had the surgery start becoming more successful and we were starting to implant windows. We kind of got the basic imaging, but we haven't like fully gone to see how much layers of the cortex we can identify.
Okay. Any other questions? All right. Well, thank you. Very nice talk, Raj.
Thank you.
John Francis.
All right. I'm just going to share my screen real quick. you all see my cursor as well?
Yes, we do.
Okay, cool. So hi everyone, my name is John, and I will be talking about my ongoing thesis project for the Robert D. Clark Honors College that investigates the relationship between cholinergic and noradrenergic activity and behavioral state in mice. So to begin with some background, an animal's ability to successfully engage with a task is largely variable. However, it has been shown that there is an optimal arousal state under which an animal can perform optimally. So that's seen in the Yerkes and Dodson curve here, where we have increases in arousal from low to medium levels increases performance, but further increases in arousal from medium to high levels degrade that performance. So arousal encompasses both brain state, which describes the physiological activity of the brain, and behavioral state, which describes the observable behavioral patterns and movements of the animal itself. So moment-to-moment fluctuations in this brain state and arousal ultimately influence an animal's ability to successfully perform various perceptual or cognitive tasks. So this behavioral assay here allows us to investigate the relationship between these changes in behavior and their underlying neural circuitry through a careful analysis of external behavioral motifs, such as walk velocity, whisker motion, and pupil diameter. And since various behavioral metrics have been shown to be closely related to cortical activity in the brain, as shown in this figure, we can therefore utilize these changes in external behavioral motifs, such as pupil diameter, to partially explain these changes in behavioral and arousal state and their underlying neural circuitry. So these moment-to-moment changes in behavioral state are partially influenced by various neuromodulatory systems within the brain, as seen in this figure. So neuromodulators are neurons that modulate the activity of other neurons. And so varying activity within these neuromodulatory systems ultimately produce changes in an animal's cognition or sensory processing by projecting to various parts of the brain. So the two neuromodulatory cell types of interest within this study are cholinergic and noradrenergic neurons. Cholinergic neurons produce and release the neurotransmitter acetylcholine, and noradrenergic neurons produce and release the neurotransmitter noradrenaline. So as seen in this figure here, cholinergic projections from nuclei in the basal forebrain and noradrenergic projections from nuclei in the locus coeruleus innervate to various target regions in the cortex, which ultimately influences cognition and sensory processing. So this background sort of forms the basis upon which I'll actually conduct my project. So my project will investigate this relationship between cholinergic and noradrenergic activity and behavioral state in three ways. So first, we'll confirm this relationship between neuromodulatory activity and arousal by including additional behavioral motifs in our analyses, which includes whisker pad motion energy and tail motion. Second, we'll determine whether changes in neuromodulatory activity precede or follow the onset of behavioral events. And third, we'll determine if acetylcholine and noradrenaline neuromodulation across multiple cortical regions is temporally related. So to accomplish these goals, we must first visualize cholinergic and noradrenergic activity in awake mice. So we're able to visualize axon activity by injecting DBH Cre mice in the locus coeruleus and chat Cre mice in the basal forebrain with GCAMP6 virus, which is a fluorescent calcium indicator that allows these neurons to fluoresce under microscopy when active. So this virus will project from these nuclei to the surface of the cortex, ultimately allowing us to actually visualize their fluorescence. So after being injected, head posted, and having a cranial window installed, we then perform two-photon axon imaging and behavioral data acquisition to directly compare axonal activity to observable behavioral motifs. So these external metrics of behavioral state include pupil diameter, snout motion, whisker pad motion energy, grooming, tail motion, and walk velocity. And then some mice will also be sacrificed to perform histological verification of injection sites to ensure that we're actually imaging from cholinergic or noradrenergic neurons. So my preliminary data indicates that after performing the histological verification of injections, we can successfully observe fluorescence in cholinergic or noradrenergic neurons. So this figure in the left here is actually showing noradrenergic cell bodies, and the figure on the right is showing cholinergic cell bodies. These are both taken from the cortex. And additionally, after performing two-photon microscopy, while concurrently recording behavioral variables such as pupil diameter, whisker and snout motion, as seen by these green boxes here, we found that changes in behavioral state are related to observable changes in neuromodulatory activity in the cortex. And that brings us to these traces down here. So this delta fluorescence over baseline fluorescence trace, or DF over F, shows that we see an increase in cholinergic fluorescence at around the same time that pupil diameter, walk velocity, and whisker motion increase. So these set of traces on the right here are kind of like a magnification of this sort of region in here. So here we see an increase in axon, acetylcholine, or cholinergic axon DF over F, as we see pupil diameter increase, walk velocity increase, and whisker pad motion increase. So these data ultimately support previously cited literature showing that changes in cholinergic neuromodulatory activity do indeed reflect changes in behavioral state. So future work includes analyzing the temporal relationship between neuromodulatory activity and the onset of behavioral events. Additionally, we're also going to be we're going to be determining if there exists a temporal relationship between the neuromodulation of multiple cortical regions. So I'd also like to acknowledge my mentors, Lindsay Collins and David McCormick, and thank you all for listening.
Okay, great. Thank you. I can open it up now to any questions. John, what's your what's the density of labeled cells when you look at it histologically in the locus coeruleus and in the basal brain? Is it sparse or just cells? Because your picture shows here, 6 cells on the left and maybe 10 on the right. How dense was the labeling?
Yeah, so I believe that these are AI-162 mice. So these are transgenic mice. So these images were actually taken from, these were injected mice. In AI-162 mice, we see much denser labeling. But in the injected mice, we do see some labeling, but it can be somewhat sparse depending on if the injection was successful or not. But in this case, it looks like the injections were successful. So it really depends on whether or not the injection was successful. And if it was successful, you would see dense labeling, like the picture on the right compared to the picture on the left.
Okay, any other questions? We have time now. We have a few minutes for general questions or questions about any of the other presentations, if anybody wants to ask one. Well, if there are not any questions, so I'm going to close the session. I want to thank you all for excellent presentations, and I know it's all a little difficult not seeing the high resolution posters, but I want to thank you all for the effort you put in, and I hope we all get back to research soon and are able to continue on with your projects. And I know we're opening up right now, Julie, so hopefully soon you'll be back in the lab and able to give a real life presentation next time. All right. Thank you all. Have a good evening and take care.
Thank you.
Thank you.
Bye.
Bye. Great job, everyone.